On chapter 6, jump into the gene pool of survival of the sickest. And this chapter is mainly about the interactions between viruses, bacteria, vaccines, and the effect and changes that they have made to our genes. The first ever vaccine ever created was actually just cowpox spores, which provided protection from smallpox. Additionally, evolution has been shaped by adaptations to viruses and bacteria and the integration of viruses and bacteria. So our first vocab word is antigenic drift, which means when a mutation occurs in the DNA of a virus or when the virus acquires new genes from a related strain, which is what causes outbreaks in pandemics. And as much as 1/3 of our cells are actually made up of viruses. And next, virals theory, or persisting viruses, are viruses that have migrated into the human genome over time and now work with the human body towards evolution in a symbiotic relationship. And the viruses get a permanent home in the genome, while humans can borrow some of the virus's code that can be used for mutations, which can possibly help us evolve more efficiently than if we didn't.
If one gene fails in its coding, then another can just pick up in its place. Only around 3% of our genes create and build proteins, while about half of the 97%, which were initially called junk DNA and are now called non-coding DNA because they are not directly responsible for making up proteins, are considered jumping genes or transposons. Transposons are nomadic mutations of genes that relocate in certain parts of a genome and cause mutation to aid in survival and evolution. Stress increases the activity of jumping genes. Transposons are descendants of viruses, such as retrotransposons from retroviruses, which help us evolve much faster. They are very active in the first part of the brain development. Malloyum says that brain diversity can be part of this. McClinton believed that in circumstances, the whole sequences of DNA can be moved from one place to another to trigger a significant change in the genome, which are jumping genes or transposons. Mutation was an accident, but natural selection occurs when the accident was helpful. Mutations can be caused by out-of-place nucleotides, radiation, powerful chemicals, or the sun. Retroviruses are viruses made of RNA, like HIV, and use an enzyme, reverse transcriptase, transcribe themselves from RNA into DNA. They can literally change your DNA. Today, retroviruses make up 8% of a person's genome and have found a permanent place in our DNA called HERVs, or human endogenous retroviruses.
Weismann had the germ plasma theory, which divides the body cells into two groups, germ cells and somatic cells. The Weismann barriers stand between germ cells and somatic cells. That theory claims that information in somatic cells is never passed on to germ cells. So mutation that occurs in somatic cells, or on the somatic side of the Weismann barrier, cannot move over to the germ cells and will not be passed on to the offspring. However, mutation in germ cells can affect somatic cells and our offspring because all instructions have genetic code originating from germ cells, from the parents. The barrier isn't completely impenetrable, as retroviruses or viruses can penetrate and carry DNA from somatic cells to germ cells. Hypermutation is bacteria's ability to adapt rapidly by a massive increase in mutation rate. Overall increase in mutation suggests that a genome has the ability to order mutations on demand, where the regular genetic program isn't good enough for survival and reproduction. Lastly, the Lamarck theory of inherited traits, which believed that traits acquired by the parents throughout their lifetime can be passed on to their offspring. This was debunked by Darwin and now has gained some credibility and transformed into the study of epigenetics, which is the study of heritable changes in gene expression that do not involve changes to underlying DNA sequence.